I am Dr. Tomas D. Morales, President of Cal State San Bernardino. Welcome to the 2025 and 5th Annual Grad Slam, showcasing the outstanding research and creative activities of graduate students from all across the campuses of the California State University. Participants take up the challenge of communicating complex research and data to a non-specialist audience in 3 minutes or less. Grad Slam, or 3-minute thesis competitions, take place all over the world and have become commonplace to recognize graduate student achievements. CSUSB is proud to be joining San Jose State, Bakersfield, San Diego State, and Long Beach, and having the opportunity to serve as a host for the Slam. I would also like to say, on behalf of all the CSU presidents, thank you to our students, competitors, for your commitment to excellence and the time you have all put in towards this competition. Please know we are incredibly proud of the students of the CSU for your cutting-edge research. I am grateful that the Grad Slam competition affords us the opportunity to recognize the innovators we have studying at our CSU campuses. As I have said over and over again, education is the key to securing, growing, and sustaining the economic and overall health of a region. As an anchor institution in the Inland Empire of Southern California, that is our focus here at CSUSB, and that goes beyond undergraduate education. We need to support the education of those seeking to pursue advanced degrees to become future educators, researchers, and leaders. We cannot move forward without access to opportunities for research that follow rigorous scientific protocols and practices. The role of graduate students in higher education, let alone the specific work each is doing, is important to who we are as a region, a state, and a nation, and a world. A special thank you today to the Chancellor's Office, along with graduate and associate deans throughout the system, for making the event possible, and to everyone serving as a judge or a moderator. Thank you. Good luck to everyone competing today.
Our next message is from Dr. Brent Foster, the California State University Assistant Vice Chancellor for Academic Programs and Faculty Development.
Hello, everybody. Are you hearing me okay?
We do. We hear you.
Okay. Well, thank you for the opportunity. And I want to start by thanking San Bernardino and our colleagues. This is just a tremendous amount of work to put these together, and it has been an absolute joy to see it coming to fruition today. So thank you for allowing me to be part of this. I also want to thank and echo the affirmations of our contestants today, and those that have continued to come throughout the day and applaud your success. Congrats on this achievement, and congrats on your soon-to-be graduations as well. It's just a real honor to be part of this excellent event. Now, my PhD is in communication with a focus on radio, TV, and film. And as you might imagine, I've spent a lot of time in broadcast studios. And if you've ever been in one, you know about the red light, sometimes referred to as a key light or a tally light or just a straight up on-air light. It's that small bulb that turns on when you're live. There's typically a countdown, a flutter in your stomach, and then just a glowing dot that means you're on. And we have a saying in the broadcast world, timing is everything. That is literally true even down to the second, much as it is for you today. When I worked in radio, if I created a 30-second commercial, it was expected to actually be 28 or 29 seconds, because if all commercials were exactly 30 seconds, there'd be no transition time, and ultimately the commercial break would run long. That little red light, it taught me something important. Yes, timing is everything, but also knowledge means nothing if you can't communicate it. And that's what makes today's competition so powerful. You all have the knowledge. You've put in the work. You've got these great experiments and observations and data and results. And now, in just 3 minutes, you get to light up your own version of that red bulb and show the world why what you do matters. Now, not everyone in this Zoom room speaks the language of labs or literature reviews, but everyone here understands passion, story, curiosity. And today, that's exactly what you'll bring. Thank you and good luck to all of you.
Thank you so much, Assistant Vice Chancellor Foster. I join President Morales and Assistant Vice Chancellor Foster in welcoming all of you to today's event. I especially welcome all of our student participants with us here today, as well as their family, friends, and mentors. We're so glad to have you here. I also want to welcome all of our colleagues and campus representatives from throughout the Cal State system, as well as the Friends of California Higher Education that are here today. It's wonderful that you could join us. And last, but certainly not least, I am delighted to welcome our judges. So our first judge is Dr. Jerry McMurtry. Dr. McMurtry, please turn on your camera and say hello.
Hello, everyone. I'm looking forward to hearing some amazing presentations, and thank you all for the hard work that you've done.
Thank you for being here today. Dr. McMurtry is Dean of the College of Graduate Studies at the University of Idaho and President of the Western Association of Graduate Schools. Our second judge is Trustee Jean Picker Furstenberg. Trustee Furstenberg, please turn on your camera and say hello.
Hello. I'm very glad to be here today and most interested in these presentations because communication is magic. And when you can do that, you can be very successful. So thank you for letting me participate today.
Thank you for being here with us today. Trustee Furstenberg served as president and CEO of the American Film Institute for 28 years and currently serves on the California State University Board of Trustees. Next is Joseph Kirschbaum. Mr. Kirschbaum, please turn on your camera and say hello.
Hello, everyone. It's a real privilege to be here. I'm really looking forward to participating in this event, and please, all of you, keep reaching.
Thank you for being with us today. Mr. Kirschbaum is a CSUSB alum and currently serves as Director of Defense Capabilities and Management at the U.S. Government Accountability Office. And finally, we have Vanessa Wiarco. Ms. Wiarco, please turn on your camera and say hello.
Good afternoon, buenas tardes, everyone. Pleasure to be here representing KVCR Public Media, and excited to hear all of your student 3-minute thesis presentations. As an alum from Cal Poly Pomona, I'm really excited to be part of this, and I look forward to being a judge and seeing who comes out as number 1 and then 2 and 3, because I think we have 3 categories of awards. So congrats, and look forward to hearing from you all.
Thank you so much for being with us today. Ms. Wiarco is the Community Engagement Manager for KVCR Public Media, the Inland Empire's PBS, NPR, and FNX stations. We're thrilled that you all are joining us as judges for this 5th annual CSU system-wide Grad Slam competition. I would like to introduce the moderator for the rest of the finals, Ms. Lacey Kendall. Lacey Kendall is a seasoned broadcaster with over 30,000 hours of live on-air experience, including roles as news director, program director, and general manager for an NBC News radio affiliate. She has covered natural disasters for NBC, was an engineer for the recent Blind Spot series on the History Channel, and currently hosts Public Radio International's Education Insight, funded by Growing Inland Achievement. As an educator, she's taught broadcasting, communication, public relations, and journalism courses at San Bernardino Valley College, the University of La Verne, and Cal State San Bernardino, where she established the Coyote Radio and Advertising Lab, the Pause Radio and Multimedia Learning Lab at Palm Desert, and the XReal Immersive Learning Library in Palm Desert. Finally, Lacey is the only person in history to have been honored with Cal State San Bernardino's Outstanding Employee of the Year, Outstanding Lecturer, President's Team Award, and the Do Good Award for service to the community. Wow. We welcome Lacey Kendall.
Thank you, Dr. Vickers. I really appreciate being here. And I very much appreciate Grad Slam for so many reasons. But what I value most is its role as a creative platform to highlight the incredible work of our talented graduate students. As we all know, the CSU takes pride in offering outstanding graduate education, consistently supporting our students and empowering them to reach exceptional levels of expertise through research, scholarship, and creative activities. I too am a graduate of CSUSB's Master of Communication Studies program. And I'm certain that, like me, everyone here today has enormous pride in their university's graduate programs. Recognizing the importance of graduate education, my colleagues and I across the CSU campuses are working to support research and graduate education because they're so closely intertwined. It's essential that the CSU continues to prioritize and support research and graduate education, recognizing the mutual benefits that come from the ongoing growth of both. We all understand and value the significance of graduate education, increased earning potential, specialized expertise, and other concrete advantages that come with that advanced degree. Just a couple of hours ago, we all enjoyed preliminary round presentations, and now we're excited to move forward with the top 2 scoring students from each round and present the top 10 finalists who achieved the highest scores and who will advance to the final round of the competition. Let's applaud these 12 impressive finalists. They earned their spot in the finals after excelling in the preliminary competition earlier today as representatives of their campuses. Reaching this stage was a challenging accomplishment, and we commend all of you for your hard work and your commitment to your field. Congratulations once again, and we eagerly anticipate your presentations this afternoon. And now we'd like to take time to review the guidelines and the rules of this afternoon's session. Each of our participants today has been allowed to use only one slide to complement their presentation. During the competition, participants will be judged based on their ability to successfully engage a non-specialist audience while communicating key details about the research in 3 minutes or less. The judging criteria for today are available for you in your event guide. As you can see, the judges are looking for clarity, organization, language appropriateness, significance, delivery, visuals, and engagement. However, students must follow that strict 3-minute time allotment, which is a remarkable achievement, to say the least. Competitors who go over that time allotment are disqualified. It's a daunting task. Despite being an online event, each competitor will be presenting live, not via a recording. You are in for a real treat this afternoon. So before we begin the competition, we do have a few housekeeping notes to share with you. We know you'd all like to cheer on your participants, and chat is open for exactly that purpose. Please be respectful to all of our participants while doing so. The way we'll proceed for each student today is that I will introduce the student who will then deliver their presentation. Afterwards, while the judges tally their scores, I will chat with the student to learn a little bit more about them. Please note that this chat has no influence on the scoring. It's just for the sake of a little chit chat while we're waiting for the results. After all students have presented, the judges will submit their scores and winners will be announced. We'll have a first place and a second place winner based on the judges' scores, but we'll also have an Audience Choice Award winner based on your votes. Information will follow on how to participate in the Audience Choice selection after the students present. Okay, I'm sure that our students are ready and eager and probably a little bit nervous to see them. So let's begin. And our first participant is Ava Besecker. Ava, would you please turn on your camera and your microphone and say hello?
Hi.
Hello. Thank you. So while the Ava, first of all, let's talk a little bit about you.
Sure.
So what inspired your interest in this particular research topic?
I've always fell in love with the ocean and specifically corals. They're not going to be around for much longer. Scientists predict that they're going to go away by 2050. So I felt an urgent need to do this. And I study abroad in the Galapagos in undergrad, and I fell in love with it. I met my partner there. It was such a great experience that I had to go back there for my master's thesis.
Okay. What would you say is the biggest misconception that folks have about your research?
Ooh, I feel like a lot of people think that the Galapagos are untouched. It's mostly nature there, but actually a lot of people travel there every year. Hundreds of thousands of people go there. There's lots of civilization, and I think people just think that it's relatively untouched.
Very good. So are you ready to go?
Yes.
All righty. Ava Besecker, you're on.
In 1835, Charles Darwin traveled to the Galapagos Islands when he witnessed iguanas that could swim in the ocean and birds with different shaped beaks based off of what they ate and where they lived. These observations laid the foundation for Darwin's theory of evolution by natural selection. Thousands of people travel to the Galapagos every year to get a glimpse of these fascinating creatures. But what if I told you that there's a lesser-known hero in these same islands, one that's fighting for its life beneath the waves? I'm talking about coral. Not as cute as a sea lion, maybe, but their survival impacts your seafood dinner, your beach vacations, and even the air you breathe. In the 1980s, a mass bleaching event wiped out 90% of all reef-building corals in the Galapagos, with many more bleaching events preceding, leaving just 10% of all Galapagos corals remaining today. The corals remaining in the Galapagos represent populations adapted to endure extreme ocean conditions, different from the typical environmental conditions found in other tropical coral ecosystems. When you combine extreme ocean conditions with slow-growing corals, it results in a very slow recovery for many species, but with the toughest corals surviving. These survivors are the most temperature resilient in the Galapagos. In 2022, the first coral nursery in the Galapagos was established using fragments from these strongest corals to restore the decimated reefs. Therefore, for my research, I use this coral nursery to better understand why some corals are able to survive in warmer waters and others are not. I analyze coral genetics and growth, which give us clues about how well corals can handle these warmer temperatures. I do this by two main methods: the use of 2D photos that stitch together using computer software to create 3D models of coral called photogrammetry, as you can see in the top right of the slide with a growth model of one of my corals. Secondly, I use genetic tools to look closely at the DNA of corals. This is important because some differences can't be seen just by looking at the species. They're only visible on the genetic level. By identifying individuals with the best ability to survive warming temperatures, my research informs methods for creating climate-resilient coral nurseries worldwide. Evaluating coral populations already exposed to environmental conditions predicted for the next few decades will provide crucial insights for global coral reef conservation. Darwin only had the opportunity to explore what was above the water in the Galapagos, but never had the chance to dive below the surface. I am picking up where Darwin left off, uncovering the importance of Galapagos corals in one of the most fascinating places in the world. Thank you.
Thank you very much, Ava. And while the judges are completing their scores, I wanted to learn a little bit more about you. Tell us what you love most about your campus and your graduate program.
So I'm at CSU Monterey Bay. I live right next to campus. I live about 5 minutes away. And campus is located only about a mile from the beach, so I'm able to walk there in the evenings. I love to go on top of the dunes and be able to see the sunset. I feel like that is the best way to decompress after a long day in the lab or just a long day mentally. And so I love to go to the beach. And I also have the opportunity to study at Moss Landing Marine Labs. It's one of the best marine labs in the country. And I just feel so lucky to be able to step into a place where just truly amazing research is going on.
Okay. What has been a highlight of your graduate career so far?
I attended my very first research conference, Western Society of Naturalists, in November. I had never been to a conference before, so it was really cool to just go from talk to talk and learning about what everybody was up to. And it was a really great networking opportunity to learn about all the different research going on the West Coast.
And finally, what's next for your research after this competition, Ava?
So I'm currently writing my thesis manuscript, so I'll work on continuing that, continuing data collection, and then hopefully next spring I will graduate and defend my thesis after my third year.
Okay, very good. It seems the judges are still pondering. So I wanted to ask you if you could collaborate. Think about this for a minute. If you could collaborate with any expert from another field, what might that be? Who might that be?
Any other field? That's tricky. Could I does David Attenborough count? Because I know that he is
Totally.
technically science, but I feel
Mhm.
like he's technically science communication and media. I
Mhm.
just think he's so legendary, and watching documentaries growing up was the way that I fell in love with the ocean. And so I would love to collaborate with him in the future.
All right. And if you weren't working on this topic, what other topic would you have chosen?
Ooh. Inside of marine science or outside?
Your choice.
Okay, let's see. I've always found psychology super interesting, so I feel like maybe doing some sort of neuroscience, psychology on potentially marine mammals would be cool. I think that would be a really interesting field to go down.
Very good. Okay, I'm checking to see if our judges are finished. I still do not have an approval. So what is the most challenging part of the research you've conducted? Looking back on that, what will you always remember as the real tough spot in this master's thesis?
Lab work is really, really time intensive. I feel like there are always things that go wrong that you don't anticipate. And so
Mm-hmm.
if I have been in the lab all day for 7 hours, something could completely go wrong and I would have to go back into the lab the next day and redo it. So I feel like
Gotcha.
I did not anticipate long hours sitting in the lab while I look out the window and see it's nice and sunny outside when I could be outside, but instead I'm stuck in the lab. So those days are really hard. It's really rewarding to get good results back, though, I will say.
All right. Well, Ava, we appreciate you joining us here today, and we will move on to our second contestant. Good luck.
Thank you.
Mm-hmm. All right. And our next contestant is Colin Kubek. Colin, would you please turn on your camera and your microphone and say hello to us?
Hey everyone.
All right, we can hear you. Are you ready to go, Colin?
Yep.
All right. Here we go.
Picture this. You're a California strawberry grower. You've been loaned millions of dollars to prep and plant this field, and before you can even harvest the first fruit, a severe infestation of spider mites threatens to wipe out your crop for the entire year. You're used to spraying, but now the rules are changing. The Department of Pesticide Regulation has introduced a sustainable pest management roadmap that aims to take away key conventional pesticides that growers have relied on for decades. So, what's the plan B? That's what my research asked. Growers are frustrated. Many of the organic products they're sold just don't work. And as that pesticide toolbox shrinks, they're looking for efficacious solutions, not just another green label. So I studied UVC light, a technology often touted as sustainable, but with little real data to back it up in pest management. UVC is a specific band of ultraviolet radiation that works by damaging the DNA of living cells. I studied UVC on two major arthropod pests of strawberries, the two-spotted spider mite and lygus bugs. In the lab, I exposed all life stages of both pests to controlled dosages of UVC using this chamber seen in the bottom left here. In the field, I collaborated with Trick Robotics to use this R2D2 on steroids looking robot seen in the bottom right. And the results were astounding. In both lab and field trials, UVC killed nearly all spider mite eggs. For the juveniles, it showed moderate efficacy, and for the adults, it did not work. For lygus bugs, it also did not work for any life stage, showing it's not a viable management strategy for that pest. These findings are huge because very few pesticides work on two-spotted spider mite eggs, and resistance is a growing issue. Strawberry growers are already using my data to improve their pest management program by applying UVC earlier in the season to target spider mite eggs. UVC is used to control powdery mildew also, a major strawberry disease. So finding out that it works on mites makes it a disease and pest management tool, compounding its costs and furthering its case for adoption. In agriculture, there are no silver bullets, and UVC is no different. It has to be applied early and preventatively in order to be effective against mite eggs. But if done right, UVC has the capacity to replace many pesticides in the California strawberry industry. So the next time you go to a grocery store and buy a clamshell of strawberries, think of the science and the light that went into protecting them. Thank you.
And thank you, Colin. Before we move on, we're going to chit-chat to learn just a little bit more about you before our judges say that they have added up their scores and whatnot. I wanted to ask you what inspired your interest in this particular research topic?
The sustainable pest management roadmap that I mentioned is going to be in place by 2050. And if effective management strategies for these pests aren't found before 2050, it puts us as Americans at a huge food security risk because we would be unable to produce the same amount of food that we're able to.
Yeah.
With these conventional pesticides. So finding effective and sustainable alternatives is what really motivates me.
Okay. What's the biggest misconception that people have about your research?
I think a lot of the pest control advisors, which are the people in California who look over the agricultural crops and make sure they protect them from the bugs and diseases. A lot of pest control advisors that I talk to say that, oh, UVC doesn't work because it didn't kill the adults. And they'll just have it run over once, look at the leaf and say, oh, it didn't kill the adults. But I just try and get the point across that it works on the eggs. So if you apply it early, then it'll be much more effective than if it's a curative treatment once there's already lots of adults in there.
Yeah. You said you're in a hurry to make the findings and this information available to these folks. So what is beyond that? What is next for this research? Where does it need to go next?
Finding so some biological control, some beneficial mites also are used to control these spider mites. So finding out if UVC is compatible with these beneficial mites could really create a potent and effective management program for spider mites. So figuring out compatibility with different beneficial insects would be the next step.
Very good. Well, thank you for chit-chatting with us here today, Colin. We appreciate it. It appears the judges have entered their scores, and so we're ready to introduce our next competitor, and that would be Peter. Peter, would you please turn on your camera and your microphone and say hello?
Hello.
All right, great. We can hear you. Are you
Mhm.
ready to go?
Yes.
Excellent.
Start.
Then let's go.
Limb loss is an unfortunate reality for over 57 million people around the world. Many rely on artificial limbs, which often require manual actions like pressing a button just to close their hand and grab something. But imagine being able to move a robotic arm only by thinking about it. For people whose lives are deeply affected by limb loss, that kind of technology could be truly life-changing. My research focuses on decoding muscle signals to make such devices possible. At the core of this technology are neural machine interfaces, or NMIs, systems that allow a computer to communicate directly with the human body. Signals from the body are sent to the computer, which interprets the person's intention and then moves the robotic arm, what we call a prosthetic. Our neural machine interface relies on bioelectricity, the tiny electrical impulses our brain uses to communicate with muscles. You can follow along with the bottom part of my slide. We pick up these signals using a few stickers applied around the circumference of the forearm. And while this concept isn't new, what we've invented is a novel way to decode these signals using a neural network, which is an AI that functions similarly to a human brain. We made it compact enough to run on a microcontroller no bigger than 2 fingers. Such microcontroller can be powered with AA batteries, making this technology truly compact and portable. So how did we do it? Let me fill you in on a few technical details. We used the electromyography arrays to record a subject perform motions that cover basic hand intentions. With that data, we trained a small convolutional neural network. The magic is in the fact that the neural network was initially designed to process voice commands, but our practice shows that it works extremely well on bioelectrical signals. Then we deployed the resulting algorithm to a Sony microcontroller and had our subject perform the gestures. Our algorithm showed 90% accuracy in detecting those gestures, and it made predictions in real time. So what does this mean? We are the first ones in the world to create a miniature, fast, and accurate muscle decoding system that fits on a tiny microcontroller, creating a sign language for intuitive prosthetic control. However, the impact of this work is much deeper. Beyond helping those with limb loss or impairment, our neural machine interface technology has broader applications in areas like human assisting robots, stroke rehabilitation devices, and even video game and VR systems. In short, neural machine interfaces have the potential to transform how we interact with machines and shape the future of human-machine integration. Thank you.
Thank you, Peter, very much. While the judges are completing their scores, I'd like to ask you a little bit about yourself.
Of course.
In your view, what has made graduate research important to society?
Well, I think the best way to learn is to learn by doing,
Mhm.
right? Uh, so therefore, in order to learn how certain things work, you should do them. If you want to learn how research works, you should go out and perform research. And there's no better platform to perform research than and learn how to do research properly than graduate school, especially in a place like SF State where there's a lot of undergraduate and graduate research, and it's very well funded, and it's it's a great place to be.
Is there an unexpected skill that you gained while on your research journey?
You know what's interesting? My whole research journey in general was pretty unexpected. I wanted to become a software engineer a long time ago when I was in my undergrad, and I did a couple internships, and then I realized that maybe it's not something for me. But I've come to understand that I really love experimenting and just building things that are new that solve certain problems. And I think it's more so not a skill, but rather a realization that I'm more interested in finding problems and creating solutions for them is definitely rather than just being a part of something that just perform some small function. I think that's the biggest thing that I've gained out of
All
my
right.
journey.
Yeah. And tell us what you like most about your own campus or your graduate program there.
I really love the flexibility of my graduate program, because you might have noticed I'm actually a biology master's student, but this research project is more more so an engineering project. You know, Performing interdisciplinary research is something that um, I think this is probably the best thing that my graduate program has allowed me to do. Give me the flexibility to combine multiple fields um, yeah,
Yeah,
multiple fields. Yeah,
very good.
big
Well, best
computer.
of luck to you, Peter.
Thank you.
It appears the judges have entered their scores, and so we're ready to introduce our next competitor. And our next competitor is Sunena. Sunena, if you could, please turn on your camera and your microphone and say hello, and let me know that I pronounced your name correctly.
Yes, you pronounced it correctly.
Very good. Well, welcome. Are you ready to go?
Yes, I'm ready.
Okay, whenever you're ready, dear.
Did you know that sepsis is a leading cause of death in hospitalized patients? Imagine this. You have a loved one who is sick. At first, they have a sore throat, cough, and fever. A few days later, they tell you they feel short of breath, and you notice they're more confused. You take them to the hospital, and the doctor tells you they suspect sepsis. Now, you may be wondering, what is sepsis? Well, sepsis is a life-threatening condition in which your immune system overreacts to an infection. This overreaction leads to widespread inflammation and damage to multiple organs. When the doctor suspects sepsis, they'll order blood cultures. Early diagnosis and treatment are crucial to improving outcomes in patients with sepsis, and blood cultures represent the gold standard for diagnosing bloodstream infections. As you can see on the slide, there's a picture of two bottles. Those are blood culture bottles, and they contain a liquid in them so that when the patient's blood sample is added to it, it allows bacteria to grow. That way, we can identify which bacteria is causing the infection and target it with the right antibiotics. A problem can occur, however, if proper protocols are not followed when collecting blood cultures, as this can lead to contamination. Blood culture contamination is related to poor patient outcomes, such as overuse of antibiotics, which leads to antibiotic resistance and delays in adequate diagnosis and treatment. Several factors can contribute to blood culture contamination, such as insufficient skin cleansing or touching the skin after disinfection. Another factor that research has found is deep skin residing bacteria, which is what the picture on the bottom middle is showing. As you can see, there are small particles of bacteria that can enter the catheter of your needle when the patient is pricked. If you connect your blood culture bottles directly to that, it'll suction up the tiny particles of bacteria into that blood culture bottle, leading to a false positive result. This is where my research comes in. My research aims to decrease blood culture contamination rates by utilizing a waste tube, which we call the Z-tube, as you can see on the far right side of the screen, to catch the initial 1.5 to 2 mLs of blood, thus eliminating that skin-residing bacteria before it reaches the blood culture bottles. At San Joaquin General Hospital, we implemented the Z-tubes in March and April. In February, we had a contamination rate of 2.8%. In March, we've been able to decrease that with our Z-tubes by 50%, from 2.8% to 1.3%, and even further in April to 1.1%. This data supports the fact that hospitals can utilize waste tubes to decrease contamination rates and lead to better patient outcomes. Once again, early diagnosis and treatment can significantly improve the chance of survival in patients with sepsis. So God forbid, if your loved one is diagnosed with sepsis, you want to rest assured that their blood cultures will not be contaminated so they can be treated adequately. Thank you.
Thank you very much, Sanena. While the judges are completing their scores, again, I'd like to ask you a little bit about yourself. First of all, could you tell us the thing that you love most about your own campus?
What I love about my campus is that it's on, well, my campus is on program is online, but we do get together once a year for like 2 days. We do like an immersion where we get together with our cohort and our teachers and everything. And I love that everyone's very welcoming. Our professors are very understanding and very supportive. And I don't really feel like that disconnect from being on an online program.
Mhm.
A lot of,
Good.
yeah, I feel like there's a lot of closeness and with my cohort and with my professors and everything.
That's always so nice when you're working on your master's degree. What would you say is a surprising discovery that you made while working on your thesis that you didn't expect to run into?
There was several things. For one, I haven't had experience with research. So I didn't know like all the aspects that go into it. Like working with the leadership team, there's like so many different people you have to interact with to make a project successful. Because you could have a great idea, but if you don't have like the right buy-in, like the right people aren't on board, it'll make it difficult to continue forward with your project.
Mm-hmm. What's the most challenging part of the research that you ran into?
I would say that the most challenging, first of all, I would like to say that everyone was very supportive in my hospital, like on board with the project, which was very helpful. I would say the hardest part was probably figuring out like the timeline because my graduation is next week. So like we had a shorter time frame to do like the research, but that's one thing that I was grateful for my professors that they allowed like the deadlines to be a little bit stretched.
Mm-hmm.
Finished my report. I finished the data at the end of April and I submitted my paper yesterday, even though I'm graduating next week. Yeah, I like the flexibility of being able to stretch it further, so get those 2 months of data. 'Cause I feel
All right.
like 1 month they would have been less data to go off of.
Yeah. Well, good luck to you. And we thank you, Sanena, for joining us. While the judges are, well, the judges have actually got their scores in. So let's move on to our next competitor. And that would be Adrienne.
Hi, everyone. I'm Adrienne Cuevas. Thank you for introducing me.
Yes, we're happy to have you. Are you ready to go?
Yes, one thing I'd like to note, as I did in the preliminary, for some reason my other advisor was cut off, so I also would like to extend a warm thank you to Dr. Adrienne M. Valadez as well to show credit where credit is due.
Very nice. Nicely done.
Thank you.
All right. Then, Adrienne, if you are ready, we're ready.
I want to make sure that we're all on the same page. That was a pretty extraordinary circumstance. Like I said, we'll go through background and method, which is how we did the study, findings, and implications. So first thing is setting the stage and making sure we all know what non-binary means. As I said, we define it as people who identify outside of the male/female gender binary. This can include folks who identify as both simultaneously, as one or the other at different points in time, or as something completely else. Why do this research? Well, the first literature I reviewed was 2024 United States Census Bureau found that approximately 1.1% of adults self-identify as non-binary. And 1.1 may seem like a small number, but in actuality, that amounts to nearly 4 million adults in the US. Secondly, a lot of gender and sexuality research has found that these are two interactive aspects of identity, but very little of this research is done on this unique population, who has a very unique relationship to non-binary and gender. How did we do it? We used semi-structured interviews via Zoom, and we found our participants using online forums and snowball methods, which is where our participants were able to recommend other folks who identified similarly. We used thematic analysis, which is a type of analysis for qualitative research, meaning research that doesn't use numbers. We transcribed our interviews, and using multiple raters, we found common patterns and themes that build our coding book. The codes that we found are here in my findings and implications columns. So first was self-conceptualization, which is how each participant explains the concept of their gender identity for themselves. The implication of that is that we found that identity labels and how non-binary relates to other identities varied based on personal experiences, meaning each definition was extremely personal to each individual participant. Next, we have arrival to gender identity. Participants describe the processes by which they came to their gender identity. And we found that this process is an ongoing consideration of both internal and external influences. Internal meaning their own personal thoughts and feelings towards their identity, and external meaning the way they walk through the world and their relationships. Finally, we have bidirectional influences, which is participants' understanding of gender can shift their understanding of sexuality and vice versa. We found that in alignment with other research, gender and sexuality were interconnected. They informed each other and brought new information that changed the broader self-concept. In a time where marginalized populations are further getting persecuted, it's more important than ever to uplift these stories and voices. Thank you so much.
Thank you, Adrienne. Good recovery there.
Thank you so much.
All right. While our judges are completing their scores, I wanted to ask you a little bit about yourself.
Okay.
What part of this research excited you the most while you were working on it?
We were able to actually have a pretty large range of age in our participants. A lot of research today is primarily made up of college age participants because those are the people who are looking at these studies. But we actually had multiple participants who were over the age of 50. So we were able to capture their rich and unique experiences as the sociocultural, political climate around queerness has changed.
Mm-hmm. So how do you stay motivated when you're going through challenging portions of the research? Because we know, all of us who've gone through a master's program before, we know that this thesis can be quite daunting. How do you handle it?
Absolutely. Well, on a human level, I'm really lucky to be supported by my family. I'm a first generation college student, and so there's a lot of love and support from my family, who are incredibly proud of the research that I'm doing.
Mhm.
And in terms of the research topic, I'm very lucky to live in an incredibly diverse neighborhood where you're meeting people from different walks of life, and these stories have a richness that inform the human experience and how we understand each other. So everyone I meet who gets inspired by this research just continues to light that fire myself.
Yeah. Well, thank you so much for chatting with us today, Adrienne. We appreciate it. Best of luck to you.
Thank
And
you
it
so much.
appears that the judges have entered their scores, so we're ready to meet our next competitor. And that would be Amanda. Amanda,
Hi. Oh, sorry.
hi. No, you're fine. You're fine. So welcome. Are you ready?
Yeah.
Okay. Whenever you're ready, we are too.
Okay. I'll wait for the timer.
Okay.
Hello, everyone. My name is Amanda, and my project is the development of non-opioid therapeutics for chronic pain. If everyone pinches themselves right now, they will feel pain. Usually, that pain is temporary, but if that pain lasts longer than 3 months, that is defined as chronic pain. In 2023, the United States National Center of Health reported that 24.3% of adults have chronic pain. In addition, 8.5% adults have high impact chronic pain, which means they are limited to daily activities. There are current medication, and the most prescribed are opioids. But when opioids are used long term, it could lead to adverse side effects such as addiction, tolerance, and death. According to the CDC State Unintentional Drug Overdose Reporting System, in 2023, 81.4% of overdose deaths involve at least one opioid. Therefore, there's currently an unmet medical need to help treat chronic pain. So that's where my research comes in. So my research is to develop a non-opioid therapeutic for chronic pain. So how do we do that? So in our human body, we have enzymes that help break down stuff. FA and SEH are enzymes. What they do is that they break down the green shape, which is pain relief, and turn it into the red shape, which is no pain relief. So we propose that if we can make a drug that will block both FA and SCH, we believe that we can prevent making those red shapes that we don't want and overall have pain relief inside of our body. As a result, we were able to find a drug 4D that has shown to block FA and SCH to provide pain relief. In the bar chart on the right-hand side, we can see that it took a much lower dosage of our drug to have the same amount of pain relief as ketoprofen, which is a known pain reliever on the market. So overall, we are able to find a drug that will provide pain relief in rat models. So where are we right now in the drug development process? Well, we're in the beginning stages. We do drug design, so we make a bunch of drugs, and then after that, we choose the best drug out of all of them to send to our collaborators at Cal State East Bay, who will then do rat testing to see if our drug provides any pain relief. If that happens and we do a bunch of more animal testing, then we can move on to human clinical studies. And if that works out well, then we can do FDA approval, and then our drug can be distributed to the general population for monitoring and safety to make sure that our drug has no adverse side effects. So where are we in this project? Well, 4D is actually not our best compound. We actually need to optimize the left-hand side of our drug to make a better drug that will block FA and SCH in pain relief and be an oral drug. So again, my project is to develop a non-opioid therapeutic for chronic pain. Thank you.
Thank you very much, Amanda. So while the judges are completing their scores, once again, I'm going to ask you a few questions to find out just a little bit more about you.
Yeah.
Tell us what you love most about your campus or even your graduate program.
Yeah, so I did my undergrad here at Cal State Fullerton. I transitioned over as a grad student. What I really love is my department, the chemistry and biochemistry department, because we're such a small community that we're able to empower each other and support each other through difficult times. And there's a bunch of resources at Cal State Fullerton, which I really love, like UROC. So it's really a great way to empower my way to do research as a grad student.
Yeah. What inspired you to pick this specific topic?
Yeah, so as you can probably hear about the opioid epidemic and all the scary statistics, you can see that that really shows that there's a lot of unmet medical need for chronic pain. So therefore, that kind of drove me to join this lab because we're doing like actual work that is impactful. And overall, I want to improve human health. So joining this lab really kind of motivates my work.
What would be one piece of advice you'd give someone who's interested in the very same field of research?
Yeah, I would tell them to try everything, and keep always working hard because you are making an impact no matter what, and always strive to be your best.
All right. And what's your favorite activity when you aren't studying or working on a thesis?
Yeah, my favorite thing. Well, I struggle with work-life balance, but if I do have a off time, I usually love to go to cafes. My favorite thing to drink is matcha, so I really love exploring the Orange County area since I'm from LA.
Yeah, sounds great. Well, thank you so much for chatting with us, Amanda. We appreciate you. Best of luck. And it appears that the judges have entered their scores and we're ready to introduce the next competitor. And that would be Sarah. Sarah, would you please turn on your camera's on and your microphone? Say hello to us.
Hello, how are you?
Very well. Thank you for joining us here.
Thank you for having me.
Are you ready?
Yes.
Okay. When you're ready, we are.
Thank you. Hello, everyone. My name is Sarah Holman de Bruin, and this topic is personal to me. I have been a long-term caregiver to both my parents, one of which has been living with HIV since I was 3 years old, diagnosed back in the early 90s when silence, stigma, and fear were everywhere. Growing up, I saw firsthand just how isolating it is when nobody talks about sexual health, especially if you don't fit the mold of who's supposed to be at risk. That experience shaped everything I do now. It's why I chose to focus on older adults and sexual health because, let's be honest, people are still having sex as they get older, but nobody seems to want to talk about it. Most people are shocked to hear this, but STIs, or sexually transmitted infection, rates in adults 65 and older have more than doubled in the past decade. Yet they're not a part of STI prevention efforts. And here's what really struck me. The CDC, or Centers for Disease Control, has an STI guideline that is 180 pages long, yet they don't mention older adults, not even once. So if the CDC isn't acknowledging this population, is it really all that surprising that providers and the rest of us aren't paying attention either? That type of invisibility has dire consequences. Because of this, I decided to survey 89 older adults between the ages of 50 to 100 in Southern California's Inland Empire. I used a validated STI knowledge questionnaire so my findings could build upon further research. I also added additional questions, bringing the total to 33, so participants could then score up to 33 points depending on how many they had correct. And what I found was pretty eye-opening. The average score of knowledge was just 19 out of 33. 86% said their providers had never discussed STI testing with them, and only 43% had been tested for an STI in the past 2 years. Those within residential care, so take nursing homes, they scored lower than those living independently. And those that weren't sexually active and also believed in common misconceptions, so like condoms are only used to prevent pregnancy, they also scored lower. But there is some good news. Those that were sexually active and didn't buy into those common misconceptions, they tended to know more. And these patterns I found weren't just random. They accounted for more than 40% of the differences in STI knowledge across my sample. And to me, this says one thing: we're not too late. There's opportunity here. We just need to start showing up for this age group, training providers to ask those hard questions, challenging outdated attitudes, and including older adults in those hard conversations. So why does all this matter? In one case study, a man in his late 70s tested positive for an STI. He needed to inform 3 months worth of sexual partners. He had 16. Yes, 16. He wasn't ashamed though. He was shocked. No one told him that this could happen to him. He thought STIs were just for younger people. And honestly, most people do. That's why my research is so important. Because growing older, it shouldn't mean disappearing, especially when it comes to health, especially sexual health. Because after all, what is age but a number? Thank you so much for your time.
And thank you, Sarah. So again, the judges are completing their scores right now, calculating and whatnot. And so I wanted to take a minute just to ask you a few questions about yourself.
Awesome.
Tell us what you love most about your campus or your graduate program.
Oh my gosh, I have so much good things to say about CSUF. I am a alumni from there, so I got my bachelor's in public health. And honestly, if I was being truthful with everybody, the reason why I went into my master's program is due to my mentor, Dr. Sammy. She pushed me to do more with my life and really pushed me back into academia. And I love the professors at CSUF. They are the best, and I recommend this program to anybody.
How do you believe your research could potentially change the way things are done if everyone were aware of what you just shared with us?
That's a very interesting question. I think there is a lot to change. I don't know how we would change it. That will take more research, and that's why I want to do more research. I think we need to really think about how people cognitively are aware as they get older to see how they learn better. I've always thought about how people learn differently. I'm a very visual learner myself. I'm dyslexic, so it's hard for me. And whenever you grow older, your mind starts slowing down, right? That's the natural body process. So I think just doing more research, finding out more would be so helpful.
All right. And if this were not the topic you were working on for your thesis, what would be?
Wow, that's a really good question. I am super passionate about sexually transmitted infections, but I think it would be more on community-based research, specifically within Santa Ana. I think Orange County and Santa Ana is a very weird dynamic, for a lack of better words. And so I probably would be doing community research, as it really intrigues me. And I feel like once you're in the community, you can really make an impact. And I think that I speak for everybody that's in a master's program. That's all you want to do. You want to change lives. You want to bring your knowledge to the forefront of the people.
Sure. Well, good luck to you on that, my friend.
Thank you so much.
And thanks for chatting with us, Sarah. It appears the judges have entered their scores, and so we are ready to introduce our next competitor, and that would be Monica. Monica, please turn on your camera and your microphone and say hello.
Hi.
Hi. Are you ready to go?
Yes, I'm ready.
Okay, we're ready for you. So whenever you're ready, go right ahead.
All right. Valley Oaks, sorry, I'll wait for the timer. Valley oaks are native and endemic to California. They are widespread throughout the state, and they're large organisms, so they provide a ton of important resources to the many organisms within their ecosystems, such as the endangered California condor. However, unfortunately, as a result of climate change, many valley oak populations are declining. We are seeing that adult trees are dying off, and populations are struggling to recruit enough seedlings to maintain their population size. And many researchers attribute this to drought effects. Here on this map, you can see a drought index in California as of April of this year. And I have an arrow pointing to Kern County, where I am, which is in the southern San Joaquin Valley. And the darker colors indicate higher levels of drought. And as you can see, there is quite a bit of drought here. And researchers project that valley oaks are expected to undergo population declines here specifically. So active restoration becomes really important. And that is what I am doing for my project. I have planted 2,400 acorns across 8 plots along a creek at Tejon Ranch Conservancy, which is located in the southern San Joaquin Valley. And as part of my project, I wanted to understand what factors are limiting these seedlings, and are there any active restoration methods that can be used to help them overcome this? So I found 3 different active restoration treatments that I'm going to be testing on my seedlings this year. The first one being nanochitosan. This is a biopolymer derived from chitin, and that's found in many different sources, such as different insects and fungi. And these are common enemies of plants. So plants have actually evolved to be able to sense chitin, and in response, they will upregulate their defenses. And the defense that I'm particularly interested in upregulating is their drought tolerance. As for the second treatment, this is mycorrhizae. And this is a type of fungus that can form symbiotic relationships with root systems. And under certain circumstances, this can actually allow the plant to uptake greater amounts of water and nutrients. As for the third treatment, this is something as simple as plot location. I will be testing upland versus lowland site of planting. And the idea behind this is lowland areas can act as hydrologic refugia. In these areas, water runoff can settle and accumulate in these areas and actually percolate more into the soil, allowing for greater soil moisture. And so I'm hoping that the results of this study will provide much greater insight into the factors limiting seedling survival. Thank you.
Thank you, Monica. And while the judges are completing their scores, once again, we're going to chit-chat with you just a little bit, find out about you. What has been one of the greatest highlights of your graduate career so far?
Ooh, I would have to say getting to present at the ESA conference in 2024. That was very exciting. It's a very large ecology conference, and I just felt so honored to be able to share my work there and be heard by fellow ecologists.
Yeah. So let me ask you this. If you could pick someone out, use your imagination here. If you could share your research and have a discussion about it with any figure out of history, who might that be?
Ooh, that's interesting. I feel like it would have to be someone like John Muir, because he had a big influence in America's park systems, and I feel like he'd be interested to know what's happening with our lands today and how climate change is affecting it and what we're doing about it.
Yeah. What part of this research excited you the most?
Ooh, I think getting to do research on the Tejon Ranch Conservancy, it's just so beautiful out there. Such a large piece of land, over 200,000 acres, and I just feel so lucky to get to see everything there.
And what do you do? What is your, let's say, your favorite activity when you're not studying? Because obviously off time is just as important when you're working on something so elaborate.
For sure. Well, I like to try different coffee shops around town with my fiance, and I also like to play different video games such as Minecraft. It's kind of like getting to be out in nature when you're not actually out in nature, so it's pretty fun.
Okay, very good. And it looks like our judges have made their decisions. So thank you very much for joining us, Monica, and best of luck to you, dear.
Thank you.
All right. And our next competitor up is Heather. Heather, would you please turn on your camera and your microphone and say hello?
Hello, can you hear me?
We sure can, and we're very excited. So if you're ready, we're ready to go too.
I'm ready.
Okay.
Good afternoon, everyone. My name's Heather Collins, and I'd like to take you to a different environment for the next few minutes. Imagine standing on the edge of the Sacramento-San Joaquin Delta, surrounded by winding waterways that have sustained life for thousands of years. For California tribes, this land is more than just a resource. It's an ancestral home, deeply connected to their identities. For the last 200 years, this critical landscape has been reshaped by levees, dams, and agriculture, its natural rhythms altered, its traditional stewards often sidelined in discussions about its future. My thesis asks, how can we build meaningful and lasting collaborations with tribes in resource management? As one of the most ecologically important regions in our state, efforts are increasing in the Delta to address climate change, water shortages, and habitat loss. But engaging tribes in these efforts isn't as simple as just sending an invitation. My research explores the barriers that exist to this work, with a primary focus on agency and researcher perspectives. To examine these barriers, I conducted qualitative ethnographic interviews with researchers from the California Department of Water Resources, academic institutions, and environmental organizations. I conducted informal interviews with tribal members engaged in this work to better understand their perspectives and experiences. I utilized participant observation, attending land stewardship workshops and restoration events, to witness in real time interactions between agencies and tribal representatives. And finally, I'm leveraging GIS analysis to visualize how this landscape has changed over time. Now, data collection is ongoing, but key themes are emerging in this work. Bureaucratic structures create barriers to meaningful tribal engagement, with rigid agency policies leaving little to no room for flexibility. Funding mechanisms misalign with tribal priorities, often emphasizing short-term outcomes over long-term relationship building and knowledge transfer. And finally, different tribes just have different structures and goals, meaning that a one-size-fits-all model is rarely going to be appropriate. These findings highlight the need for more adaptive, relationship-centered approaches. If we truly want to collaborate with tribes, we have to do more than just fill out the forms and check the boxes. We must move beyond compliance-driven engagement and seek to build relationships that are rooted in trust, reciprocity, and most importantly, respect for Indigenous sovereignty. It is time for us to listen, to learn, and to honor the knowledge of this land's traditional stewards. Thank you for your time today.
Yeah. Thank you, Heather. The judges are again completing their scores, so I'd like to ask you a few questions about yourself.
Sure.
What about this inspired your passion so much?
I've been really passionate about Native and Indigenous studies for most of my life, I think. I have Indigenous heritage myself. My family is very disconnected from those roots. And anthropology was kind of a way for me to understand that disconnect, to contextualize it in today's world. And so working on issues of environmental injustice or other injustices against Native and Indigenous communities has been a passion through my undergraduate work till now.
Yeah. What's the biggest misconception that folks may have about your research topic?
Yeah, I think one of the biggest misconceptions that surrounds these kind of themes is surrounding the land back movement, right? I think there's a lot of misconceptions that, you know, this is just a land grab and everyone's going to get removed from their homes. And there's a stigma that surrounds this. In reality, Native and Indigenous communities want to reconnect with their land and they want to steward their land in the traditional ways that it was stewarded. And that doesn't mean anybody needs to leave, right? They want to do that in partnership in many instances. And it's just providing the space for them to be able to do that.
Yeah. Heather, what would you say is the greatest thing about your home campus?
Oh my goodness. I love Stan State. And I think probably, I might tear up, but the most important and most impactful thing has been my faculty support. I wouldn't have made it this far without my thesis committee. They've been with me, some of them since my undergraduate degree. And I have never known a support like this. I'm very grateful for it.
Yeah, that's wonderful. Well, we appreciate you. And thank you so much. The judges are done making their tabulations, and we're going to move on and meet our next competitor. And coming up is Tracy. Tracy, would you please turn on your camera and your microphone and say hello?
Hi, everyone.
We're very happy to have you. Are you ready?
Yes.
Okay, Tracy, when you're ready, take it away.
Hi, everyone. I'm Tracy, and I'll be sharing my project on what impacts the well-being of Filipino-American caregivers. In many Filipino-American families, caregiving is woven naturally into daily life, influenced by a value called pakikisama, or the drive to keep harmony within the family. Many Filipino-Americans step up to care for their elders, often without being asked. But here's the catch. Putting others first again and again can take a huge toll on caregivers' own health. And because of pakikisama, many caregivers hesitate to seek help, even when they're overwhelmed. In research, Filipino American caregivers are often invisible. Studies talk about Asian American caregivers in general, but not the unique experiences of Filipino Americans. That's why we launched this study, to listen directly to Filipino American caregivers and find out what predicts their quality of life. We surveyed 213 Filipino Americans living in California who are actively caring for an older adult. They answered questions about how burdened they felt, how they were coping emotionally, and how much stigma they felt about seeking mental help. We ran a predictive analysis. Basically, we looked for patterns, and we found that 3 main things kept showing up: caregiver distress, negative stigma around getting mental health support, and increased perceived burden. What does this mean? When caregivers feel weighed down, judged, and alone, their health-related quality of life suffers or goes down. This is where occupational therapy comes in. Occupational therapists, or OTs, help people build healthier, more meaningful lives. OT meets people where they are, in their routines, their relationships, and their values. With the recent Medicaid amendment, OTs in California are officially recognized as mental health providers, allowing us to offer behavioral health services to caregivers. OTs can provide wellness strategies and tools to support caregivers' health, identity, and competence. OTs respect traditions like pakikisama, but also recognize that no two people experience culture the same way. Even within shared culture, every caregiver's experience is different, and OT embraces that. So when we create these interventions, we want to make sure that it's culturally tailored to address the unique challenges each Filipino American caregiver is facing. Ultimately, this research fills a gap. It paves the way for occupational therapy services that don't just offer help. They offer respect, dignity, and deep understanding for Filipino American caregivers who have been quietly caring for their families for generations with needs, hopes, and dreams of their own. Thank you.
Thank you, Tracy. The judges are completing their scores now, so I'm going to ask you a little bit more about yourself. First of all, what inspired your interest in this particular topic for your research paper?
Yeah, so our research professor did a lot of research on Asian American caregivers, and me, along with 2 of my classmates, found this super interesting, and we wanted to look into it more further, and we saw that there's just a lot of research in Asian Americans, but there's never any research on the split amongst the demographics within Asian studies. And so my 2 partners are also Filipino American, so we decided to let's focus on something that ties closely to us to make it as impactful, important for us to pursue this research.
Mhm. If you had unlimited resources, how might you expand this research? Where would you take it from here?
Yeah, so our study was only in California. We would love to expand this across the country as, I mean, the study I keep saying Filipino Americans, but it is really just focusing on California, and we would like to expand on that. On top of that, for our results, we saw in our demographics, we saw a lot of male participants. About 60% of our participants were male, which is very surprising as we thought that we would have more female participants. So that would be something really amazing to look into, especially with a lot of research coming out on mental health for males.
Mhm. All right. Well, we wish you the best of luck on that, and we appreciate you joining us. The judges have completed their scores, and so it is time to move on to our next contestant, and that would be Ayana. Ayana, hi. We can hear you loud and clear. You comfortable? Feel ready to go?
Yes.
Okay. Watch for the countdown, and when you're ready, you go right ahead, dear.
The ocean is getting hotter and fast. Marine heat waves, once rare, are now becoming stronger, longer, and more frequent. These extreme temperature spikes don't just warm the ocean, but they disrupt entire ecosystems. Take our California kelp forests, for example. They are one of the most biodiverse habitats in the ocean, and you can see that in the healthy kelp forest image. Kelp is thriving, and healthy urchins are coexisting within. To the left of that image, you'll also see a heat map from before major marine heat wave strikes, displaying stable ocean temperatures in green. But when a marine heat wave occurs, you can see dramatic increases in ocean temperatures of up to 5.4 degrees Fahrenheit higher. Now, I know that doesn't sound like a lot, but it can mean life or death for marine life, highlighting just how extreme these events have become. And when ocean temperatures rise, our kelp struggles to survive. Without kelp, species that rely on these underwater forests will lose food, shelter, and stability, leading to serious consequences for ocean wildlife. One of those species that gets affected is the purple sea urchin. You can see the devastating effects of marine heat waves in the urchin barren photo. This is kelp that could not survive the heat, and leading to a surplus of starving, unhealthy urchins. But rising temperatures don't just threaten urchins by taking away their food. They impact them at the cellular level as well. Inside their bodies, there are proteins that act as tiny machines, keeping everything running smoothly. But when it gets too hot, those proteins can sometimes start to unravel and fail. And this is where heat shock proteins step in. Heat shock proteins can help cells survive stress. But if temperatures get too high, even heat shock proteins can't keep up. This is especially dangerous for the early life history stages of sea urchins. And if you look at the images of developing urchins, you can see just how small and vulnerable they could be. So, if proteins aren't working properly at these stages, sea urchins may not survive. And that's where my research comes in. I will be conducting gene expression analyses on developing purple sea urchins within the stages shown on the slide at different temperatures in order to determine at what stage of early development this heat shock protein is turned on. This will help us understand how rising temperatures affect the early life stages of urchins and whether or not their mothers can provide any protection to help them cope with heat stress. So, if young urchins can withstand heat stress, they might just be able to survive future marine heat waves. But if they can't, their populations will struggle, leading to even more instability. So, sea urchins aren't the villains in this story. They represent vulnerable marine creatures just caught in an ever-changing ocean. And as marine heat waves push ecosystems to their limits, the question is no longer whether marine life will be affected. It's who will survive and who won't. So how hot is too hot to handle? My research aims to find out. Thank you.
Thank you, Ayana. The judges, again, are completing their scores and tabulations. So let me ask you a few questions about yourself. First off, what was the most difficult aspect of this research?
Oh, this research, so the protein that I'm looking at, there is limited research on it, especially in marine invertebrates. So I feel like that's a very difficult part. But that just leads more ways for me to put more research out there. So hopefully by the end of my project, I can publish this and it can be used for other marine work and even for understanding better molecular mechanisms for these proteins.
Yeah. It sounds like there was a great deal of research that went into this. And my first thought is, how has your family responded to all of the research you've done over this past year and then your thesis?
They have been so supportive. They're even watching today. So hi, Mom and Dad. They're always like the backbone of my research. So they are fully supporting me in everything that I do. And I'm so appreciative of it, especially because they are immigrants and they come from the Fiji Islands. They were born and raised there.
Well, that's great. So tell everybody who is watching, your parents probably know this, but what do you love most about your home campus?
My home campus is amazing. I have such a tight-knit community there. All of my professors are so welcoming. All of my peers are so nice, and I'm sure they're all watching today too. It's just really supportive, and I couldn't have asked for like better people to cheer me on and always like want to help me with research or even just, support me in everything I do, even if it's not like in academics.
Okay, very good. Well, we thank you for joining us today. We wish you the best of luck.
Thank you very much.
And we move on to, I believe this is our last competitor of the day, and it is Lydia. Lydia, would you please turn on your camera and your microphone and say hello to everyone?
Hello.
Hi. We can hear you. Everything seems great. Are you ready to go?
I'm ready.
Okay. Watch for the countdown, and when you're ready, go right ahead.
There is a condition that more than half the world's population is born with, one that makes you more likely to be killed. It's not some disease, yet every 10 minutes, it claims a life. I was born with this condition. Many of you listening were too, and some, though not born with it, will still face its side effects because of their identity. This condition? Ding, ding, ding! It's the XX chromosome. Yes, the simple complication of being female can be a death sentence. Femicide, the killing of women because they are women, is a global epidemic. A woman is killed by her partner. A girl is murdered by her family in an honor killing. We may think it doesn't affect us, but in the US alone, nearly 3 women are murdered every day in homes and neighborhoods just like ours. When this happens, you may ask, well, why didn't she leave sooner? But how could she, when the hands that feed and shelter her are the very hands that strike her? To leave would be to face hunger, homelessness, and isolation. The sad truth is that economic dependence is one of the strongest predictors of femicide because it makes a woman more vulnerable to abuse and gives her fewer exit options. So I asked, can economic empowerment reduce femicide rates? To answer this, I ran a regression analysis on 32 years of US economic data and femicide rates. I focused on female labor force participation, that is how many women work, as a key measure of economic empowerment, while accounting for the income levels and the gender wage gap to assess whether women who work were actually earning enough to achieve real financial autonomy. As you'll see in my slide, my analysis uncovered that for every 1% increase in working women, femicide rates drop by 0.43%. Now, that is no small drop, as it translates to over 225 fewer deaths every year. Yes, employment gives a woman income, independence, and social ties, lifelines when the home isn't safe. So what do we do with this knowledge? We intensify the push for policies that empower women, equal pay, affordable childcare, paid parental leave. And this isn't just left to the lawmakers alone. No, you and I have a part to play. Hire her, mentor her, align yourself with people and platforms that prioritize her safety and independence. Women's lives are at stake, and for so many, earning a living could just mean earning their lives. Thank you.
Thank you, Lydia. We appreciate you. Best of luck to you. And that concludes our competitors' presentations today. That was terrific. I hope all of you in the audience enjoyed watching the students' great presentations as much as I did. It's going to take a few minutes for the judging team to verify that all scores have been entered correctly so that we can announce the winners of this final round. So at this time, I'd like to tell you how you can vote for your favorite presentation, what we like to call the People's Choice Awards. It's a Zoom poll that will appear on your screen for you to make your selection for the People's Choice Award. Polling will be open for about 3 minutes. So please look for the poll on your screen and be sure to vote. A first place, second place, and audience choice winner will be selected, and each comes with a fabulous prize of its own. All 3 winners will receive a trophy that will be mailed to them. The first place winner will also receive a check for $1,200. The second place winner will receive $800, and the audience choice winner will receive $500. The 7 other participants who earned their spot in this final round will receive a plaque acknowledging their great achievement, and that will be mailed to them as well. So while we wait, please enjoy some highlights about our CSU campuses. All right. I believe our judges have completed all of their tabulations and voting, and we have some winners to announce. Let's see here. Let me get to the People's Choice. Okay, very good. So the People's Choice winner is the very first winner we have to announce this afternoon. And could we get a drum roll? The People's Choice winner is Ayana Dia Singh from Sonoma State. Congratulations. All right. And if everybody's ready, the second place winner is, let's have a drum roll, Sarah Holman de Bruyne. Woo! Okay, and now we're at the moment we've all waited for. Our first place winner in the California State University System Grad Slam 3-minute thesis competition. Our winner in first place is Lydia Duranguma. Lydia Duranguma is our grand prize winner today, the winner of the Grand Slam. Now, if our winners would like to activate their cameras and wave to be acknowledged, we'd love to see you. Please do so right now so we can all congratulate our winners.
Hello. Thank you so much.
Yeah. All right, and there they are. Very well done. Very, very well done, ladies. So congratulations to everyone. And before we conclude, I want to take a moment to acknowledge the very large number of people across the entire CSU system that came together to make this great event a reality. First and most importantly, thanks and congratulations to all of our student participants. Your deep comprehensive understanding of your subject coupled with your energy and your grace, have made all of us so proud to be associated with the CSU system. I want to express my heartfelt appreciation to our esteemed moderators and judges as well, whose invaluable presence and expertise have made this event possible to begin with. A huge thank you to our preliminary round judges as well. Thanks also to the members of the event steering committee who spent months planning this Grad Slam. We're also very grateful to the Chancellor's Office for sponsoring this event. A huge thank you goes out to the logistics magicians who pulled everything together. This was a hardworking team of people, but I especially wanted to thank Megan Canali and Thin Lee for going above and beyond the call of duty to make this event a success. And finally, thank you to the Chancellor's Office and CSUSB for providing funding and support for this event. We look forward to seeing all of you at next year's Grad Slam. Thanks again, everyone. Congratulations to all of the winners, and everyone have a wonderful day. Thanks for joining us.